So in this video, we're going to think a little bit about protein biochemistry, and what we're really going to focus on to start with are the amino acids, which you should know are the building blocks to proteins. And of course, proteins do all sorts of interesting stuff within cells. So proteins can be enzymes, they can be transporters, they can be structural proteins, they can be signaling proteins, anything you think of, it's almost certainly a protein that's doing it. So we really need to understand how proteins work. Okay, so in order to do this, we first of all need to understand the amino acids, which are the building blocks of proteins. So you might know that the common structure for amino acids is this. So we have a central carbon atom, we have a carbon, double bond oxygen, and an OH, which is known as a carboxylic acid group. So that's where the acid bit of the name comes from. On the other side, we have N and two hydrogens, which is the amino group. So those are the two main groups that give the amino acid its name. We've also got a hydrogen attached to that central carbon, and then we have a group that is known as R. You'll see that, you might have seen that before. So that's a hydrogen, so the R group what that R means is this is different for each of the different amino acids, and we'll think about what those different R groups are in a second. Okay, so for all of the amino acids, they've all got a central carbon. And notice that that carbon is attached to four different things. It's attached to amino group, which is an NH2 group. So we can summarise the structure down as this. So sometimes we'll see it as C. Rather than drawing out every single bond, we've got two hydrogens and the nitrogen, so that's the amino group. We've got the hydrogen, we've got COOH, we've got the carboxylic acid group, and then we've got the R group. So sometimes we'll abbreviate the structure to that. Okay, so for our amino acids, of which there are 21 amino acids, slightly depends on your definition of which ones are used biologically, but for all of them, we've got central carbon, we've got hydrogen, we've got an amino group, we've got a carboxylic acid group, and we've got the R group, which is the variable group. So each different named amino acid has a different chemical group on that R position there. Okay, so let's have a look at what some of these R groups look like in some actual amino acids. Let's start to populate this with some real biochemistry. So the most straightforward amino acid we have is glycine up the top here. So glycine, it's the full name of the amino acid, so that would be GLY in 3-letter code, or just G in the single letter code. So glycine, let's have a look at the structure. So it's got the NH2, that's the same. It's got the H, it's got the COOH, and for glycine, it's R group. So I'm going to highlight the R group in blue. Let's go for. I'm going to highlight it in orange for each one. Okay. So the R group for glycine equals just a hydrogen. Okay, so it's just a hydrogen. So in fact, glycine, you bonded to two hydrogens. The carbon, central carbon's got the normal hydrogen, and it's got an extra hydrogen there. Okay, so the R group is very simple in a glycine. Okay, it's just a hydrogen. Okay, and then that and that's actually not very chemically reactive. Hydrogen can't do very much in there, okay? So there's no particular chemical bond that hydrogen can form with any other component in the cell. Okay, so the R group for glycine is just a hydrogen, okay? Let's go to alanine, which is ALA, or just A in the amino acid codes, okay? The R for alanine is CH3. So just go through again. We've got the NH2, we've got the H, we've got the COOH. Here's our R group. So CH3, which we should know is a methyl group. Okay. So alanine has a methyl group in this R position, okay? And that starts to inform what sorts of interactions it might be able to form with other molecules. So we know that methyl, because it's carbon and hydrogen, is a relatively non-polar sort of a group. There's another video on sort of basic biochemistry explaining what polarity is, if that's something you're unfamiliar with. But when we've got carbons and hydrogens bound together, we know that those are non-polar, which tend to be hydrophobic. Okay, which means it doesn't like water. Okay, so this, if we have CH3 as our R group, this means that this amino acid is not likely to want to interact with water. It's more likely to want to interact with fats or other hydrophobic things. So because of the R group on there, that R group has particular chemistry, and that starts to give us an indication of the sorts of things it'll be able to bond to. Okay, so let's have a look at the next one. So serine, SER, or just S. Okay, serine, again, amino group, hydrogen, carboxylic acid, that's the same with each of them. The R, in this case for serine, is CH2OH. Okay, so instead of CH3, we've got 2 carbon, 2 hydrogens, and a hydroxyl group. Okay, so that OH is a hydroxyl. Which means that we've got hydroxyl, which is a polar group. So again, look at the video on polarity if you don't understand what polar means. Okay, because the oxygen is quite electronegative, that means we've got imbalancing the electrons across there. So the oxygen will be a little bit negative, the hydrogen will be a little bit positive. We know that OH groups are polar, and that means that they are hydrophilic, which means water loving. Okay, so this group is more likely to want to interact with water than this group over here. Okay, so we might find that this is on the outside of a protein where we're exposed to water, but this is more likely to be on the inside of a protein, which tends to be a more hydrophobic environment. So we can start to make some predictions as to where things might be. Okay. So let's have a look at some other chemistry. So cysteine, so CYS, or C in our amino acids. So here we've got an interesting group in that we've got an SH, so we've got some sulfur in cysteines. Okay, so we've got a group that contains sulfur that is able to form a different type of bond, which we'll see when we think about protein structure, can form disulfide bonds. So there's a particular type of bond that we find in proteins that can only form between cysteine residues. So if you've got one cysteine near to another cysteine, those sulfur groups can interact with each other and form something called a disulfide bond. So here, the chemistry, because we've got sulfur, there's a different type of bond that can be formed. So no other amino acid can form that type of bond. Okay. Let's have a look at some more. So lysine. So the R group in lysine is more complicated. Okay. So we've got CH2, we've got that 4 times. So actually there's quite a long chain that sticks out from here. So we've got CH2, 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 CH2. It points out quite a long way. And then on the end of it, we've got an amino group. So notice that lysine's got 2 amino groups. It's got the original one and it's got this one sticking out of the side. Okay, and in the cell, to a first approximation, the specific biochemistry of this is a little bit different, but to a first approximation, actually when you're in the cell and in an aqueous environment, NH2 groups become NH3 plus. Okay, so what happens is in solution you tend to get an extra proton binds onto the amino group because it's working as an acid. Okay, it's working as a base. Okay, again, there's a video that explains how that works, but sort of in solution, we know that actually this is likely to be not NH2 but NH3 with a plus. Okay, so notice that we've got an actual positive charge on this lysine. Okay, so because it's got an NH2 group, which in solution becomes NH3 plus, we have a positively charged amino acid. Okay, so again, that will change the way that it can bond. Here's aspartate. So here's its group. Okay, so notice that we have a carboxylic acid. So we've got CH2 there, and then we've got carboxylic acid, COOOH. That was the same as over there. Okay, and again, in solution, what tends to happen is nearly all the time, so C double bond OOH would become C double bond O minus. Okay, so we've got a minus, we've got a negative charge on there. So we've given up a proton. So actually, so this would be, it actually becomes COO minus in solution, okay, which means that we've got negative charge because we've lost a proton. Okay, so if you had in a protein, if you had a lysine near to an aspartate, then they would be attracted to each other. Well, we've got one positively charged amino acid because of the amino group, and one negatively charged amino acid due to the carboxylic acid group. They would then start to be attracted to each other. Okay. The other the weirdest amino acid is proline. So proline has a very odd R group, and it doesn't quite follow the normal rules. Okay, so usually we have NH2 COOH, but in proline it's a little bit weird because we have NH, just a single H, and where that H2 should be, what we actually have is this ring-like structure whereby the R group actually physically connects back to that N in what should have been the amino group. Okay, so we haven't got sort of an R sticking out the side, it's actually a ring-based structure in there. Okay, so that is relatively chemically inert. It can't form chemical bonds. There's not really anything reactive in there, and it also affects the structure of a protein, which basically the other amino acids can kind of point in different directions. This one ends up pointing in a very specific direction when we form a long chain of amino acids. So let's have a look at how that might work in terms of the primary structure of proteins. Okay, so I've shown you a variety of different amino acids. As I say, there's 21 of them, and you can look them up online for what all the different side chains are. Okay, there isn't any. Those are the main types of chemistry. So you can be non-polar, hydrophobic. You can be polar, hydrophilic. You can contain sulfur if you've got cysteine. You can be a positively charged amino acid. You can be a negatively charged amino acid, or you can have this weird proline one. Okay. So those are all the amino acids. So those are little molecules that are found in the cell. But obviously when they're protein, then they get added together. So what happens is this. Okay, so if I draw out two amino acids, okay, CH, there's the R, COOH, okay, plus a second amino acid, okay. What can happen through the process of translation, okay, I'm just going to call this R1 and R2, okay, is that those two amino acids get linked together to form a new type of bond. Okay, so H2N, CH, there's the R group, okay, and then we form a new bond. So rather than COOH and an NH2, we end up with this structure, which is C double bond O, NH, C, and then we've got the next amino acid. Okay, so this bond here is called the peptide bond. Okay, so what we've done is to link that carboxylic acid and that amino group. And actually when we do that, we lose a molecule of water, so it's called a condensation reaction. You'll notice that we've lost one H from here, one H from here, and we've lost an oxygen from here. So we've now got a peptide bond, okay, connecting our two amino acids. Okay, and in fact, when we go into a protein chain, we don't just put two amino acids, we might put 1,000 amino acids in a very long chain. So actually what we'll end up with, if this is the next peptide bond, CHR3, and off we go, we're off to the races. Okay, so that will keep going. So in fact here I've got 3 peptide bonds that have formed. Okay, so we've got the peptide bonds that have formed. Okay, so the C double bond O connected to N and NH, those are covalent bonds. Okay, so they're really pretty strong bonds. Okay, you've got your chains of carbons and nitrogen in the middle, and then you've got your R groups are sticking off to the side. Okay, so those R groups can be any of these things we like. Okay, so if our protein was, we started off with a glycine, so we might have, so we start off with glycine, so we might have a hydrogen, then we might have an aspartate, so we've got CH2COOH, then we might have a serine, so we've got one of those. So we end up with this chain of amino acids with these R groups kind of sticking out to the sides. That means that they can interact with stuff. Okay, so if we have our if we sort of abbreviate that, okay, to be, here's our chain of amino acids. So each of these circles is an amino acid. Okay, and then we've got R groups sticking out. They tend to stick on alternative sides like this. I'm going to summarise quite a lot of protein biochemistry here, because there's some important other things that you need to be aware in terms of secondary structure, and there's other videos about that. But what we've got, if we've got a chain of the R groups sticking out, and remember each of these R groups is different, then if we've got a second bit of the protein chain, so let's just take that over there. So then we've got a second bit of protein coming in at the bottom here. So this might be another region of the protein. Okay, it's also got R groups sticking out. Like that. So we can now start to have bonds form between those R groups. Okay, so here's 2 different R groups. If they're next to each other and if they have the right sort of chemical structures, they will start to form bonds with each other. So there's an interaction with an R group. Here's another interaction with some R groups. In fact, that one can interact in 2 different ways. Here we've got another R group sticking out there. Okay. So those R groups then allow us to form different types of chemical bond. Okay. So just to be clear, the peptide bonds are what I've shown between, in terms of these strong linkages here. Okay, so that's the central carbon effectively, that's the peptide bond in these chains, and then we have the R group sticking out to the side, and they can form a variety of different interactions. So I'm not going to go through the chemical structures of each of those, but they can form the bonds and it's actually in the tertiary structure. There's other videos around about protein structure, so you might want to have a look at those if you don't know what primary and tertiary structure are. But the sorts of bonds that we can have in tertiary structure, so we might have disulfide bonds. which would be if you've got cysteine residues. Okay, so if this R group is a cysteine with some sulfur in it, and this R group is a sulfene with some cysteine with some sulfur in it, they can form a bond called a disulfide bond. We can have ionic bonds. So for that you need a positive and a negative amino acid. So if here we had lysine on one side, which we know is positive, and aspartate on the other side, which we know is negative, they could form an ionic bond. What else can we have? We can have hydrogen bonds. So if you've got polar things, so if you've got two serines near each other, then they could potentially form a polar bond. Or we can have van der Waals bonds, which again, there are other videos on the internet about what those different types are. But if we have non-polar, so if we've got alanine next to another alanine, they can't form a sort of formal bond, but they will be attracted to each other because they're both relatively hydrophobic. Okay, so the important thing to realise is, this is the core amino acid structure. They've all got the same basic structure, but the R group is variable. What chemical group you have on your R group will determine the sorts of bonds that you can form. Okay, so if you've got CH3, that's a methyl, you'll be non-polar, you'll be hydrophobic. Okay, if you've got a carboxylic acid in there, then you'll be negatively charged and you might be able to form an ionic bond. Okay, the amino acids are joined together as primary structure with peptide bonds in there. And then when we get to more elaborate protein structure, those R groups can interact with each other. And if you've got the right combination of chemistry next to each other, then you can start to form some of these recognisable types of bonds in the tertiary structure of proteins. So you might want to have a look at either the basic biochemistry videos or a video on protein structure to help you understand that a little bit more. But hopefully that's given you an overview of the amino acids and their biochemistry.